Mr. Weiss, this is Ari Monchokla, and this is my osmosis experiment with the semi-permeable membrane of naked eggs. Here, I have two controls. My first one is a now naked egg, which was a raw egg placed in vinegar with a 5% acetic acid for 48 hours. My other one is a normal raw egg. Here, I have 4 naked eggs placed in 4 different solutions for 48 hours. Let's see what they look like. My first one was placed in distilled water with a blue food coloring added. Grown slightly bigger, taken on the dark blue food coloring, and the water has become lighter blue. My next one is placed in an 8 to 10% salt solution. It's become slightly smaller and it's become a little clearer skin color. My third one, maple syrup. Shrunken quite a bit and taken on the dark blue coloring. And then my last one is placed in Sprite with an 8.6 sugar content. It's gone slightly smaller as well, and skin tone has lightened up. However, in the first 6 hours, I noticed that my Sprite egg had gone a little bit of air pockets. Thank you, Mr. Weiss, for listening to this part of my experiment, and I shall continue with weighing my eggs. Mr. Weiss, I shall continue to compare the weight of the eggs from the solutions to the naked egg control. The naked egg control weighed 100 grams. Egg 1 plus 7 grams. Egg 2 minus 4 grams. Egg 3 minus 49 grams. Egg 4 minus 7 grams. Please see the attached slideshow. It concludes my results from my naked egg experiment. Thank you for listening to my video on my osmosis experiment with semi-permeable naked eggs. Hey Mr. Weiss, this slideshow gives you my background work before the 48-hour naked egg osmosis experiment and the analysis of the outcome. This slide is about the materials I used. For the 6 eggs I took, I found the net weight of each egg and then the average weight, about 46 grams. Then I put them in beakers and topped them with vinegar. This shows the gradual change in a raw egg to becoming a naked egg. I soaked them in vinegar for 48 hours. The bubbles are the result of CO2 released, and you can see the calcium carbonate on the glass. Finally, after 48 hours, I took the eggs out of the vinegar solution. Here you can see they do not have any eggshell, but a fine membrane covers the egg contents. The acetic acid in the vinegar helped to dissolve the eggshell made of calcium carbonate and release CO2 and H2O. This revealed the cell membrane of the egg, a semi-permeable membrane which lets only certain molecules in. Here you can also see the naked eggs are larger in size compared to the control raw egg. Water has entered from the vinegar solution, which had higher concentration of water into the egg through the cell membrane by the process called osmosis. Water molecules will continue to move from an area of higher water concentration to a cell of lower water concentration until there is homeostasis or equilibrium, which means that the water concentration is the same inside and outside the cell. On this slide, I weighed the eggs and noticed that there is a distinct gain in mass because of water entering the egg. Here I have also shown that my calculation of average weight gain is 54 grams. Here I have analyzed why the size and weight have increased and how the egg lost its shell. In the next 4 slides, I have shown that I have immersed 4 naked eggs into 4 different solutions, as I have shown in my video. This is a quick view of the naked eggs after 24 hours. The largest is the blue one. All the rest seem to be shrinking. This is a view of the eggs after 48 hours. Blue is still the largest, and all the rest have reduced a little. I weigh each of the eggs, and it seems like the blue egg, which was in water for 48 hours, is the largest, increased by 7 grams, and the least was the one in maple syrup for 48 hours, decreased by 49 grams. On these last 2 slides, I analyzed the reasons of the changes after a 48-hour solution bath. Thank you for watching my presentation, Mr. Weiss.